Hello, everybody. Welcome. This is another webinar as a makeup for a workshop that was originally scheduled for for last year's ISEE conference that went virtual. Today, Dr. JEANETTE Stingone will talk to us about how we can maximize the reuse of existing environmental health data. Dr. Stingone is an assistant professor at Columbia University's Mailman School of Public Health at the Department of Epidemiology. JEANETTE, it's all yours.
Great, thank you so much. As everyone to be able to see my screen. I want to start out by saying that I'm giving this sort of mini workshop or presentation, but this work is really the collaboration between multiple institutions. So I'm at Columbia, but the data center, the primary data center is at Mount Sinai. Our collaborators are at Rensselaer Polytechnic Institute, and of course we are funded by NIEHS. So to start out, the goal of the HEAR data center, and if you don't know what HEAR is by now, I'm going to talk about it a bit in a minute, is really using data science to advance environmental health research. And so everything I'm talking about today and showing you today is really in service of that goal of developing the tools and technologies that we can use to advance how we use data in environmental health research. I'm going to start out by giving a brief background of HEAR and the HEAR Data Center. I'm going to talk about how and why we built this data repository, which we think makes the reuse of existing data much more feasible. And then I'm going to give a demonstration of the repository and let you know how you can gain access to it, which I think is why most people are probably in attendance. So for those of you who don't know already, HEAR stands for the Human Health Exposure Analysis Resource. It was formerly called CHEAR for Children's Health, but it has been expanded to include studies across the life course. This is really a great example of collective science initiated by NIEHS. So the HEAR network consists of really three parts: an exposure lab network. These are labs that have coordinated together. They run QCs and interoperability tests to ensure that when samples are analyzed in one lab, they're interchangeable with what would be potentially analyzed in another lab. That's really important for when we want to pool data. There's a coordinating center that helps manage how everything works in the HEAR network, and then there's the data center. So the way it works is, as an NIH-funded study, if I have biospecimen that I want analyzed for certain environmental chemicals or targeted analytes in biospecimen, I go and apply to enter the HEAR program. If accepted, I send my samples to one of the labs. They're analyzed for free. I'll have a link later on for people who are interested in accessing the HEAR program itself. The purpose of the HEAR data center is to help with the data that are generated by the lab, both in terms of analysis of that data, as well as integration with the existing data supplied by the study. So this sounds like a great deal, right? You have your biospecimen and you get them analyzed. The partnership part of it is that you then agree to share the data that are generated by this project after a set embargo period, okay? And so that's where this data repository comes in. We have these diverse sets of studies in the HEAR program, and we want to make that data available to people. Now, you can imagine that this is sort of a big ask, right? Because these are really diverse studies of health, particularly with HEAR. They're all across the life course. There's no prior coordination, and we never really know what's coming next. So harmonization needs to be dynamic, and this resource is constantly growing. But what this resource will allow us to do is pool data and potentially address more complex questions than we could answer with just one study. So when building this repository, what we've really thought about is, how do we take away some of the laborious work of pooling data and data harmonization, take that away from the scientists so they can focus on the work that they need to do, which is asking critical questions, thinking of creative ways to reuse these data in a way that is scientifically sound, right? We want to take away the work of, wait, this study says VAR3, which happens more than we think, right? What does that actually mean? oh, they've used 2 different coding schemes. Let me go through and harmonize that, right? That's all the work that is laborious and rote, and we know how to do, but can computers do it faster and easier for us? And that's the purpose of our repository. Okay, so one of the reasons why we want to make these available to the data available to the scientific community, and why we're hosting workshops like this so that people can see how to access our repository, is again to promote collective science, the pooling of studies where appropriate, right? Also, we can reuse study data for research and teaching purposes. And if we have time, I have a quick example that I've used at Columbia of being able to use real world data to illustrate methods to students. Finally, the HHEAR repository allows for biomonitoring data across the life course. We often think about using NHANES for this, that's our sort of go-to. But there are certain limitations to NHANES in terms of chemicals being analyzed in certain age groups. And we've actually found, I gave a presentation to some state laboratories. They were really interested in being able to use HEAR to get exposure data from lots of different geographies to be able to understand, how are people being exposed. So in order to achieve these objectives, right, our repository needs not just to say, okay, here's the data, go at it, but we need to do it in a way that really facilitates their use. And so the data are not just available, but they're fair. And I'm sure most people have heard about fair data principles before, but we've created this repository to really achieve these objectives. The data are findable. So again, we're hosting these workshops, we're making them, we're gonna make the repository accessible, like through Google searches and things like that. But also even within, it's really easy to find what it is you want. The data are accessible. That's a step above available, right? It means that when you see the data, you know what it is. There are no guesses. There are no, wait, what is this? How can I use it? It's very clear in our repository and in our supporting documentation so that people can not only access it, but use it for what they need. It's interoperable. Our repository has been built upon existing semantic technologies and semantic ontologies, and I'll talk a little bit about what ontologies are in a minute. But it means that the terms we use, the codes we use, are reused from other sources, which means when you have our data, which have been harmonized to this sort of common vocabulary, you can link to other bioinformatics resources to generate knowledge. And I'm going to show an example of that in a minute. And finally, the big thing is that these data are reusable. And so that means that sometimes things don't seem the way we traditionally think about data and data dictionaries in epidemiology or environmental health. That's because we want to advance the reusability of the data. And so we make everything so that it's not only human readable, but also machine readable. And that means that we'll be able to do more with data as we progress in developing some of these technologies. So again, all of this is just background to let you know sort of how we've created this repository and why, and then I'm going to show you how you can access the data. But again, all of this was done very intentionally so that we are maximizing how people can reuse the data within the HHEAR networks. So as I said, all of this is powered by something called ontologies. So some of you may have heard this talk before, so you're going to be really used to the ontology of ice cream. But for those of you who haven't, this is just something to make it a bit more accessible. So the definition of an ontology is a formal, explicit specification of a shared conceptualization. That's by a man by the name of Tom Gruber, who you may have heard of because he's the man behind Siri, which is in all of our iPhones in our pockets. But what does that definition really mean, this shared conceptualization and making it formal and explicit? Well, we have the ontology of ice cream as an example. If I say to you, I love ice cream, or I ate ice cream today, you know in your head an idea of what I mean. Maybe you don't know exactly what I mean, but you have this shared conceptualization of what ice cream means. You may think about the different flavors, vanilla, chocolate. You may think about different presentations, right? Served with fruit, served with cookies. But we as a whole have this shared conceptualization. If there is a robot and I want to teach that robot what ice cream is, how do I do it? I have to describe all of these key characteristics or key points or key examples, right? All of those things. And the robot's understanding, the depth of the understanding, will vary based on how granular I am in my specification of that conceptualization. So if I say to my robot or to Siri, where can I get ice cream? A very simple thing would just be, okay, a text search, and I'm going to look for all of the stores that have ice cream in the title. But that would miss a lot, right? If we don't go into a deeper specification of what ice cream is, that it's a food, maybe then it's found in a grocery store, right? It can be served in a cone, maybe that's in a cart at the park, right? You can see how the granularity we use to specify our terms will dictate how much a computer can help us achieve our goals. So I'm not developing anything to find ice cream, although if someone does, you can totally feel free to share that with me because I do love ice cream. As a researcher, though, as a perinatal researcher, I do have a shared conceptualization of my terms. So when I say I have data on birth outcomes and I want to communicate that to a computer to help me do the work that I want to do, I have to be very explicit about what do I mean by birth outcomes. I mean things like birth size. I mean gestational age. I may mean specific phenotypes like preterm or premature, which often people use interchangeably to mean the same thing, right? This slide here sort of shows how we sort of break apart a term that we all sort of have this understanding of, but makes it very explicit. Again, going from human understandable to machine understandable. And so as part of that, what underlies our entire data repository is the HEAR ontology. This provides sort of our standard vocabulary that we use to communicate sort of what the data are, right? Again, I'm speaking it in a way that's much more simplistic than what actually is going on behind the scenes. But that's because the purpose of this talk is not to go into the use of knowledge graphs and the advanced technologies that we're using, but just so people have an understanding of what goes into the repository that you're going to use. So we use this standard vocabulary so that way we have standard terms across all of the studies. And so here is just a brief look at some of the indicators we have, or the main classes of information. You can see under targeted analyte, we have a lot of different types of assays that are run in the HHEAR labs. And you can see that this standard vocabulary is grounded in existing resources. So we take an example like triclosan. Now, in our labs, the HEAR labs, triclosan is denoted with TCS. So every HEAR lab denotes triclosan as TCS, but it's grounded in an existing ontology, CHEBI, or the chemical entities of biological interest. I'm going to talk about why that's important in a minute. But I also want to talk about, let's say there's a study that had pre-measured triclosan, and they deposit their data with HHEAR. Well, let's say they've called their variable T1. How do we know that that's triclosan? Normally, we have to go to a data dictionary, right, and look it up. Okay, T1 means triclosan measured at a certain point. We do that for you. We map to our terms for triclosan. So that way, all measures of triclosan it's clear what they are and that they are the same thing across studies. Again, it's not rocket science, but it's the kind of work that takes humans time that machines can do for us and allow us to concentrate on the scientific aspects of our work. The more domain specific, I shouldn't say just scientific. There's definitely science behind a lot of this computer science work. So again, I talked about how these are grounded in existing resources because that gives us this added benefit that we can link to bioinformatics resources. So if you look up triclosan in CHEBI, now again, this is the code that we use to sort of say, okay, this is what this data value is about, right? It's about triclosan. You'll see that they have very clear definitions, and those definitions have information that might be useful to us. So again, this isn't happening right now, but we've laid the groundwork to be able to develop tools that can say, okay, these data are on triclosan. We can then go into CHEBI or other bioinformatics resources. Because we're using the same code, we can then link to the description. So if we wanted to do a search that says, show me the data about exposures from personal care products, right? We can have the computer do it for us. We've laid the groundwork for that to be done. So again, it's really exciting to be able to say, okay, we've created this repository that's going to have immediate benefits, which is the reuse of data. But on the sort of flip side, or that's the sort of B side of the record, right? That you don't know is going to be good, but is good. This idea that we are laying the groundwork for future data science initiatives to come in and help us generate knowledge using the existing data we already have, I think is really exciting. So I did want to mention it. But probably more of interest to you in the immediate future is that the use of these ontologies is what allows us to make meaningful searches across all of the studies. So you can imagine that if your question is, identify all of the studies with measurements of phthalates in mothers and has use of folic acid supplements during the first trimester, you can search our repository and answer that question. I'm not promising that the data are there right now, although actually for that one, there are data right now. But that's the kind of meaningful searches that you'll be able to do. And on the other part of using this standard vocabulary is it harmonizes variables to a common vocabulary. So that means no more struggling with what var1 is, or some of the unique variable names we as epidemiologists and environmental health scientists tend to use. No more having to recode everyone's different coding schemes. All of that has been done for you through our repository. I want to go through a brief example of how you would traditionally do data harmonization and how our ontology helps, because one of the things we've done, again, very intentionally, is left a lot of the final stages of data harmonization up to the researcher. And again, that is intentional because data harmonization will vary based on your research question. And again, we don't want to stymie anyone's science, right? We want to enhance your science and take over the tasks that are laborious and rote that you don't need to be doing, but we don't want to take away your autonomy or your creativity in your questions. So imagine you have 3 studies and they have 3 different measures of maternal education. Normally, when you wanted to go to data harmonization, you'd have to find the common denominators across the 3. What we are doing in the background in our repository is we map our ontology, map all of the terms within individual studies to ontologies. So what do I mean by that? Study 1 had 4 different levels of education. So let's just take the first, no formal education. Well, no formal education is equivalent to never attended high school, right? It's a subclass of that. If you've never attended high school, if you have no formal education, you've obviously never attended high school. If you have no formal education, you've also never completed high school, right? It's a subclass of that. What we're doing is finding where each of the individual study levels fits underneath our ontology. And then what normally you would do is find the groupings, right, that cover all of those individual study levels, right? You're finding the harmonized pieces that capture the information in the 3 studies. And so when you do that, you get one single variable for your pooled analysis. And so now you've lost some granularity in finding the similarities across the studies. Well, if your goal is to pool these 3 studies no matter what, and maternal education is a confounding variable, and you're okay with a bit of residual confounding because you want to get the sample size or the exposure variation across the 3 studies, this might work for you. But let's say maternal education is one of your primary research questions. And you might say, you know what? I'm okay with having a more limited sample size to get the granularity of education. You don't want us to force you to use this pooled variable, right? You want to be able to use the granular data in study 3 alone. And so our repository has been, again, intentionally set up that decision is up to you. We take you as far as this part, mapping to the ontology terms. And probably in the next month or so, we're going to roll out suggested programming code to go to this step, to go to the fully pooled step. But it's not going to be automatic. So that way you have the choice. And again, that's sort of the purpose of maximizing reuse, is not to create this sort of okay data set that kind of fits everyone's goals, but isn't ideal for anyone. We really want to reuse and maximize the data for you so that you get the most out of it for your analysis and your research question. And that's one of the reasons why we do this. Okay, so now to the main event. I'm going to demonstrate our repository. So just hold on with me 1 minute while I get everything set up. So you should be seeing the HEAR Data Center website right now. Hopefully you are. Yes, thank you, Arianthi. Okay, so this website is really sort of the go-to for thinking about getting some initial information about the studies. So this is just the landing page. You can see here we have our current statistics about the number of studies, how many specimens have been analyzed, the types of lab panels that have been run, as well as the types of health outcomes that have been studied. There are also, really briefly, resources that are available, presentations and publications, software packages, a description and link to our ontology. But there's also on this studies tab, a list of all of the different projects that have been accepted into HEAR Now Here. And just a note, when I say here, I'm including HEAR sort of in that. I don't usually differentiate between them. So what you can see are all of the different projects. And once you gain access to HEAR, you can download the individual study files. So these are just the files that are pretty much what the investigators of the original studies have provided, as well as the lab data that has been analyzed through the HEAR network. They're not harmonized, so these are just the raw data. You can do some quick searches. These are the projects that have currently available data. And so if you know specifically, these are the studies I'm interested in, and I just want the raw files from those studies, this is where you would go. In order to access that, you would just go to log in and register for access. And it will take you to this HEAR data use guide. There's a brief introduction. There are some terms and conditions that you agree to. This, I'm showing this because in addition to giving you access to the raw data, I'm calling it raw, but it also includes derived variables and things like that. Once you have access to that, you will also have access to our repository, which I'm going to be showing to you in a minute. But again, you agree to certain terms and conditions, so it's important that you read through these. We have a publication policy. We have, not trying to identify individuals, appropriate acknowledgements, as well as some best practices and security standards. So even though the data are available once you register, you do agree to this sort of data use agreement. So this is for, again, the data on just the individual studies. When you want access to the HEAR data repository, again, you will register for a HEAR account. Once you do that, you will receive another email from us inviting you to register for our repository. You will get a login and password, and you will come to this website. The underlying software that was developed at RPI that we use basically to power the repository is called HAT. And so that's how you see it in the address bar. But again, this address will be provided in the email you receive, and it's accessible through the regular HEAR data center website. And you will log in and put in your password, and then you will have access to the repository. So you can see I'm already logged in. I'm just going to go to the dashboard quick. This is what you will see when you first log in. You will see our dashboard. You will see there are things that you can click on, and then there are things that are grayed out. Again, as I've said before, we've intentionally made this repository to be dynamic and growing as HEAR grows. So I'm going to show you the functionality that we have now. And then at the end, I'm going to talk to you a little bit about the functionality that will be available in the next 6 to 12 months. So very exciting stuff. So you can see as of right now, how much data are currently in the repository. We currently have 4 studies in the repository. That includes 229 different variables. 88,376 different data values. And we currently are using 13 loaded ontologies. So the idea that is you want to be able to search data. Let me just actually bring you to here, to the homepage. I brought you to the dashboard. I apologize for that. This looks very similar, but it has some additional buttons, right? So in here, we provide you with just some background and just some instruction. To begin searching and browsing, you click on Search Data and Request Dataset. To retrieve your data sets and also your code books, I'll explain that in a minute, you just click here. We also have our documentation at the bottom. So if you just click on the documentation and user guide, it will take you to our documentation website. I just want to show you the documentation first, because I think that's also really helpful to have good documentation when you're using sort of a new site or any new piece of software. So again, we give you a little bit of background about the repository, and we have links to both accessing the repository and registering and creating your account. We also have our support email. Again, because we're using ontologies, some of the terms and definitions we use may not be what you're used to. So we have a whole page on the key terms and definitions that we're using, including how we define time periods across the life course, terms you may not be familiar with examples. So again, it's very clear what we mean when you look at the repository. And all of this documentation will help facilitate your use of the repository. And finally, we have a clarification of ambiguous terms. Every time we publish a new version of the repository, sort of, just not a new version, but like a new release, we will update our term listings. So you'll be able to see, I'm not sure what this term means. You have a place to look from the very beginning to be able to say, okay, this is a very clear definition. Then we have some user guides that describe different things. When I show you the download files, you'll see that, we are used to, when we get data from someone, having a data file and then a code book that tells us what the variable names mean and all of that. Rather than doing that, we store all of the metadata in the column headers for the data downloads. You'll see what I mean about that in a minute. And decoding those might not be intuitive at first, although it does. It has a set pattern, so it's easy to learn. But we have a user guide for that. We have a master code book for categorical variables, right? Again, we harmonize codes across all of the studies. And the good thing is that those codes never change. So if 110 is the code for female, it will always be the code for female. It will never change. That's really great if you have set up your code already, or you've developed certain mappings that you have in your programming of assigning labels. You don't have to worry about that changing. If you download your data today, or if you download it 3 months from now, those codes and labels will stay the same. And then we also have a complete listing of analytes, abbreviations, and common units within our HEAR data repository. So again, it's very clear what's being measured and how it's being reported to you. So that is our documentation. And if we have time at the end, I can show you sort of specifically. But again, you could just go there through the user guide and browse on your own. It's pretty self-explanatory. Okay, so let's start out with searching our data. Searching the data, this is going to just take a minute, because this search is actually really deep. It searches the data itself, as opposed to just searching metadata. So this search takes a little bit longer on the computer, and we're working on speed right now to get it to go a little bit faster. But what you trade off in speed, you're getting in depth, right? So you're able to actually search on values of the data, as opposed to just saying, does this study have information on phthalates? You can say, does this study have actual data on average concentrations of MEP during pre-pregnancy? So again, so here it is. It's not that long, but you're trading that little bit of speed for a lot of depth. So what you can see here is a visualization of the repository itself. One thing I want to point out, things are denoted in colors But if you hover over it, you see the labels of those colors. So this makes it really accessible for people who might suffer from any kind of visual impairments or color blindness. You don't actually have to see the color to get the understanding. So there are multiple ways to search the repository, and I'm going to go through them one by one. And so as I do that, I'm just going to make my screen a little bit bigger so it's clearer to see. One thing I should also point out is you can turn these different search rubrics on and off to make them bigger or smaller, depending upon what you are looking at. So the first thing here on the left is the studies search. And this is, I know about these studies beforehand from the HEAR data website. And I know that I want to get all of the data from a particular study. I can come here, I can select that and hit search. I'm not going to do that now, but what it will do is it will limit my data to just the data within that study. The other thing we have done, again, to make things really transparent and understandable, is we have differentiated data that comes from the HEAR labs with data that is supplied by the parent study. So when you see lab, that means that file comes from the HEAR lab. Those data were generated by a HEAR lab. If you see PD, that means like principal, oh gosh, what does the P stand for? It's like principal investigator derived. There's an actual meaning for this in the help document. I apologize. I just totally went blank. But basically that PD means that these are from the parent study, right? So these are maybe the accompanying demographics, the health outcomes data, and any pre-measured environmental exposures that they may have had in their parent study. So again, if this allows you to sort of differentiate what's from the HEAR labs, what's been supplied by the parent studies. And again, we list the title of the study as well as the study number. And all of these match exactly to what's on the HEAR data center. So again, you can see within each study, the types of data that are available for each study just through browsing. And so again, really quickly, let's say I'm interested in this study 34, which is about metals exposure in birth. I might select all of the study variables and then hit search. So again, this takes a couple of seconds because it's going into the knowledge graph and it's sort of saying, okay, I'm just going to find the data that is associated with this study. And so it'll just take a little bit of a minute, but I want you to be able to see how what happens when you search by study. Again, as I was talking about while we're waiting, I'll sort of talk a little bit about the second column. Oh, nope, it's always faster than I think it's going to be. So now you can see that the pie charts have totally changed. Why? Because I'm just describing the data in study 34. And it's a very clear visualization. You can see the time periods that where study 34 collected data, right? Birth and pregnancy. And you can see the types of information that are in study 34. So I'm going to clear this search just so I can show you the full things in the menus later on. But just so you can see, you can go through this sort of iteratively to say, okay, I'm interested in study 34, but maybe I'm only interested in the demographics, or I'm only interested in the arsenic in drinking water. I'm going to go over this in a minute, just how to sort of read these entities and attributes tabs. But you can go through and do these searches iteratively to really hone in on the information that you need. So I'm just going to clear this search and we will start over again. So the second, while we're waiting, the second group is called object collections and roles. And this is basically when you want to search by the who are the data about? Are they about a child? Are they about a mother? Are you interested in samples? You might wonder like, okay, well, a child, a mother, these are all people. Why are you differentiating? And you'll see in a minute, and maybe I can do this now while we wait, we indicate that they're human. Again, right now, the HEAR data repository only contains information on people, but who's to say in 3 years, we might not want to deposit toxicologic data in this interface. we are prepared to do that, right? Because by indicating, oh, the child is a human, we allow for the possibility that the child could be a mouse, right? Or some other animal that might be the subject of a study. So again, all of this is to be dynamic and growing. So again, you might say, I only want information on children. Again, all of these menus can be expanded, and you can see exactly the type of information that we have. So you can see here information on child anatomy, which maybe sounds a little strange, are things like head circumference and waist circumference. For mother, we have information on the patella and tibia. I can just let you know this is about lead measurements, and there's ways to see that when you search. We also list all of the different types of samples. So as you can see in these first 4 studies, we have 4 different types of matrices that have been analyzed for metabolites and targeted analytes. So again, it's a way to just browse and visualize the type of data that are there. And if you know what, I have a study about maternal urine, and I only want to look at other studies that have data on maternal urine during pregnancy, you can select and search and only see that data. The next group is the one probably most likely that you're going to use. So I'm going to just shut these off just so we can see the entities and attributes a little bit bigger. This is the what, okay? What are the things that are being measured? What are the variables we have in our data set? You can see that we have, let me make this a little bit bigger. You can see that everything is grouped under these, what we call indicators, and then information is nested underneath them. So for example, in the category of alcohol, tobacco, and illicit drug use, in just these 4 studies, we have data on e-cigarette use, the smoker status of the caregiver of a child, information on environmental tobacco smoke exposure of the child, and also mother smoking status. You can see, in addition, let's just look at smoking status, that we have specific data points that we report on, right? So we have whether a mother ever smoked or whether she was a non-smoker, whether she was a prenatal non-smoker, so she just didn't smoke during pregnancy, or whether she did smoke during pregnancy. So again, this idea of searching on the data itself, searching the values of the data, is sort of a step beyond than just saying, does the study have smoking information? And so you can just sort of browse through and see all of the different things that you can select on. So again, you can select on just the data you want to look at. So for example, let's say we're interested in looking at the relationship between pre-pregnancy body mass of the mother and child's birth weight. So we might go in and just select mother body mass index and all of the categories for that. We would select child body weight. We're interested in getting data on that. And let's say we want a demographic variable. We want the child's biological sex. So we will select those. That's all I'm interested in for my study. So I hit search. While it's doing that, I just want to talk a little bit about this last category, which is time. We map all time periods across the studies to basically developmental windows and windows across the life course. Why? If I do a study and I have a baseline and you have a study and you have a baseline, yes, they're both baseline, but what does that actually mean? Your baseline could be in adulthood and my baseline could be in childhood. And so we map all time periods to actual, life course stages. So that promotes harmonization across studies. And more importantly, knowing that if the time periods aren't the same, you may not want to pool, right? You may not want to pool exposures measured in the first trimester with exposures measured pre-pregnancy, right? So it's important that we keep track of time, and our repository is able to do that. So now you can see the search. You can see that of the variables I selected, they're actually present in all 4 studies. And you see that here, right? Because you'll see in the entities and attributes, only the things I've selected are shown. You will see that, again, it encompasses data on the child and mother, makes sense. And it's across all 4 studies, okay? So now I want to access these data. I want to look at them. So I go up again, back after I've searched, and I click download. And download will give me 2 options, 1 value per row versus multiple values per row. we want to select multiple values per row. And that will give us that sort of standard tabular shape to our data that we are used to, where each row is an observation or an individual, and each column is a variable. So we select multiple values per row. We then get asked about alignment. Again, this is made to be really flexible, the software that underlies our repository, again, called HHEAR. And so there's lots of different things you can do. What we found works best for us is to align everything by subject. Again, that means that each row is an individual subject, and we want all of the data for that subject in the columns. So you get one row with all of the information for that person. And again, that tends to be typically how we see our data. The next piece, but there are other options that you could play around with if you wanted to. The next option is what to do about categorical values. So again, the simplest one of biological sex of male, female. People use all different codes, 0, 1, 1, 2, MF, male, female. And so how you want to see the data, again, is up to you. You can use codes that we provide and again, generate a code book, which means we will output the codes for you to see. 110 always means female, and so on and so forth. But you may say, you know what, I've done this search before and I already have a code book. All I want to do is update the data. And you might not want that new code book because again, our codes never change. So that would be the second option, use codes without generating a code book. The last is, I just want the data from the studies, how it was shown. And that would be just use values. We're going to go to codes and generate codebook. And then for time resolution, this is if there are actual time measures in the data. We actually don't have that in our repository at the current time because all of our data are publicly available and have been de-identified. So just, it doesn't, you could just leave this and not touch it. You then hit confirm and it will start the download process for you. I have actually already done the download, but I'm going to show you what the file looks like. Oh, well, it takes you to this, which is your download screen. You can get there after you do a search, or you can also navigate here from the home page. So I've already done this search. So if you look here at the search I did earlier today, which is this December 1st, 11/24/27, you will see that there are 3 files. And so I'm sorry, I did 2 searches here. And so they're sort of, all the files are kind of mixed in together, but you'll see 2 on December 1st, this 12/1/11/24 and this 12/1/11/29. So these, I just did 2 different downloads to prepare for this. It's very easy to just download the files as they're created. And there are always 3 files created if you indicate you want a code book. There is the object alignment, which is basically, these are the data. There is the download alignment codebook, which again, very self-explanatory, is the codebook. And then there is something called sources. These are the DOIs for all of the data that you downloaded. So I'm just going to download this. You'll see it just comes up as a CSV file. Let me just drag, which one was that? 29. So I actually want this one, 24. Open this up. And I'm going to show you what this, oh, I'm sorry, I downloaded the codebook, not the sources. This is what I want. There we go. Okay, so this is what the sources looks like. Again, just going to make this bigger for you to see. Remember, we said that there were data from all 4 studies in the search that we did. And you can see there are DOIs for all 4 studies. When you agree to the data use agreement for HEAR, you agree to cite these DOIs in any publications. This helps us keep track of provenance of data and give the original parent investigators credit for the work they have done. So all of these DOIs are generated for every download file that you do. As I said, it also generates a code book, and I'm going to show you what the code book looks like. Sorry, I really shouldn't have done two different studies because it definitely messed me up a little bit. Okay, so here you can see what the code book looks like. It gives the code, the label, and then the class from the ontology. So for most people, you're just going to be interested in, okay, this is the code and this is the label, right? So whenever I see 110, I know it means female. Whenever I see 111, I know it means male for biological sex. For people, though, who might want to go a step further, we do provide the ontologic class. You'll also notice that it's in a spreadsheet, it's in a CSV, which also means that it's machine readable. So if you want to load this into your software, whatever statistical software you use, to use it to automatically create labels or formats or factor variables, you can do that very easily. So again, it's human readable and machine readable. But now I just want to show you the data file that I already have open. This is what your data file will look like after the search. So remember, we searched child's birth weight. We searched child weight. We searched maternal pre-pregnancy BMI, and we searched biological sex. So you will always have the study ID provided so that you can trace the data to the study that generated it. And you will also have an ID variable to designate individuals. You can see here, this is the biological sex. And remember, I told you about those column headers. Again, everyone names their variables different things. If you want them to be meaningful, you have to put the metadata in there. So we start out with who it's about. It's a child, it's a human. What is it? It's biological sex. Biological sex is not time varying, and so it has no time period of interest. And you can see all of the studies have the same codes, 110, 111. For anyone who's harmonized data, that's all done for you, when normally you'd have to figure out what codes did these studies use and what did they do. The second variable now we see is child human body mass index. You will see that there are units. It's very clear what units are used, so it makes it difficult to sort of mess up. I only have like 2 more minutes and then I'll stop for questions. And the other piece it has is the time period. So remember, we were interested in birth weight, but this is child weight at adolescence. So we actually don't want this. And we know not to harmonize it with the birth weight, which was actually what was of interest to us. So again, this is this is weight during childhood. And we go down to F, and we can see this body weight kilogram is at birth. And so this is the birth weight. We can now see that birth weight was reported by 2 studies, 1431 and 34. You will see that they both have common units in kilogram. We do that harmonization. So you don't have some studies in grams, some in kilograms, some in pounds and ounces, right? We'll always use the same units so that it's easily harmonizable. You can see that here. And then the last piece you can see was the mother pre-pregnancy BMI. And you can see that, other variables that we've selected get downloaded, most normally weight at other time periods, because I didn't specify a time period for that. But what I want you to see here is we have mom's pre-pregnancy BMI, again, available for multiple studies, again, with the common unit of kilogram per square meter. But one study also, let me move all this over so you can see. One study also provided it as a categorical variable. We know it's categorical because there are no units provided in the column header. So again, it's this way of making the data really usable and also clearly understanding what we're seeing, right? So if you're looking at the column header and you expect a unit and it's not there, it most likely means it's categorical. And so when you go to look at the code book that was generated, you see that here a 99 means underweight. So again, really easy to interpret our downloads and really sort of clear to know what you're doing and what you're looking at. So that is the repository. And you can sort of go through once you have an account and kind of play around with that. I just want to end with some of our upcoming enhancements that are listed here. We've got some detailed help documents. We have some that are already posted, like I showed you, but some more in detail about screenshots of the repository to sort of go through what I've done today. We're going to have additional workshops, and we're also going to post videos on our resources site so that you can watch me go through this and go through an analysis in detail to sort of refresh your memory about what I spoke about today. And then we have some other things that we're working on, including some sample code and pooled analyses. So I'm going to end here. Again, all of our new resources will be available if you just go to the HEAR Data Center website. There are links to get to our documentation site and to the repository itself. And again, I'm giving this talk presentation, but this is the work of a lot of people across a lot of different institutions, and really coolly, lots of different disciplines. So this is really sort of interdisciplinary work in order to try to enhance what we can do with the data that we have within the HEAR network. So I will stop there and answer any questions that you may have. Thank you so much for your patience and attention. So hi, Gaurav. So I'm just looking at a chat, a note in the chat. Any plans to embed statistical or machine learning analyses within the website? So that's a great question. One of the things that we want to do, and I sort of have some examples. I've been using some of the data here that's publicly available already to teach students about some more advanced methods. So this was a student who was using some of the data to learn a method of LKMR, lag kernel machine regression. And so what our goal is maybe not to embed it in the website so you can do it automatically, but for people to post their R markdowns and share them. So we'll post some that we've done to sort of illustrate methods or illustrate maybe how to use a method. And that's sort of part of the additional services to the scientific community. And so we're hoping that now that we have the data, the repository sort of out there and used, we can do that. And we also actually want to provide a place for other people to do that. So let's say you do an analysis with the HEAR data and you want to share it with the group. We want to be able to have a collaborative space so that people can say, hey, here's what I did, interested to look at what you're doing. And so, yeah, expanding to Python and like Jupyter notebooks, yes, that is also part of the plan, particularly as people who maybe are not coming from environmental health, but maybe coming from more data science backgrounds tend to use this work. We do hope to sort of post some of those resources for people. Right now, we are starting out with posting things in R and probably SAS, because those are the most used within the environmental health community. And then after that, we'll probably get to Python with Jupyter notebooks. Thank you for the questions. And if there are any other questions, you can feel free to sort of unmute, I guess, too. No questions? So then if there are no questions, but please feel free to sort of interrupt me at any point. I just want to go over again how you can get access to the repository. If you just go to the HEAR Data Center website, if you just go to the login, you will see forgot your password or register. Register will take you to the data use guide. This, agreeing to this and going through this, will give you access to the full amount of information on the HEAR website, as well as the registration email for the repository. Once you get that access, you just will click on the repository link. It will take you to this HAT web at msSM.edu, and you'll be able to log in to set up your account and then log in and access all of the things that we looked at today. Any other questions about what I covered? I know I went through it quick because we have a lot of accessibility. Oh, chat, sorry. Oh, thank you, Tara. I'm glad that you like it. Yeah, and so again, I want you all to really stay tuned, because we have some cool things that we're hoping to do with these data and making them sort of available to everyone. One thing that I forgot to show you, so really quick before we end, is that not only do we have the ability to search the data itself, but you have the ability to search the ontologies. So these are the ontologies we use in the to create our repository. If you're interested in sort of looking at the terms that we use, by going to browse metadata, you can browse the ontologies and you can search for terms within ontologies. So this is the HEAR ontology. You can see currently displaying here, and you can search for terms. And so you'll see that the term you search for gets highlighted in red, and it gives you that code. So again, this is just a way, if you're thinking about, well, how does HEAR describe things? What terms is HEAR using? You can go to this and search. It's actually a really great tool and really easy to visualize what's in the ontology. So the last question, just really quickly, is, are there omic data in this repository? So there are not omic data in this repository. The data can be connected to omic data. So as of right now, if you go for HEAR, all of the metabolomics, at least, are stored in metabolomics workbench. And so those data, you saw that there were the IDs in the data set that I showed you. The ID should, this is my understanding, allow you to link to the metabolomics data so that you can get that data from metabolomics workbench. And when you gain access to the HEAR data site, this one here, there is a link to the specific study if they have metabolomics data on the workbench. So it's designed to sort of be interoperable in that way. For other omics data, as far as I know, HEAR is no longer doing measures of biological response. So there wouldn't be, I don't think, any omics data available, but it's not in the repository. But for the existing metabolomics, you can link to it. Great. I don't know if there's any last minute questions. I'm so glad that you all seem to like it and want to explore it. Please go ahead. We will be, if you register in the next few days, just wait a couple of days, you'll get an email soon. We're just sort of fine tuning that process. And then you'll be able to access and search. And please feel free to reach out to me or to the support document that we have to give feedback, right? We really want you all to explore and then be able to give us feedback. And so last but not least, since there's 1 minute left and no questions, some of the group are on this call. So Sophia Bengoa is on. She is at Mount Sinai, and she is works really hard in sort of doing a lot of the semantic mapping. Deborah McGinnis, I see, is on, and Paolo Pinheiro from RPI, sort of Paolo's the lead guy behind Hat Attack, and he can answer all of those questions for you. I don't know if Liang and Chip and Shirsha are on. They're from Mount Sinai, from our scientific computing, but they're also, again, this is really a group project and really collaborative interdisciplinary work at its best. So I think that's it for time. Thank you so much. And again, please feel free to reach out with questions.
Thank you very, very much, Jeanette. This was fantastic. And thank you all for attending. And we will see you all soon. Thank you. Bye, everybody.